Hi, now let's continue what is biomedical engineering by examples. So now we'll discuss about systems biology. Before talking about it, I want to introduce you the concept of cell culture or tissue culture. So when you think about bioengineering or life science, you can kind of come up with like growing cells, and this picture might be familiar with you. So what is tissue culture? So when you go to the laboratory, you can find some place called tissue culture room or cell culture room. Inside, there's like culturing of the cell will be happening. So this culture, tissue culture means the growth of tissues and cells that are separated from an organism. Tissue culture commonly refers to the culture of animal-derived cells and tissues, while we call specifically cell culture is growing and maintaining cells under controlled conditions. Here, can you guess what it is? The answer is usually close and ex vivo. Ex vivo means outside of a living body. So if you look at this, this is called a Petri dish, and this pinkish liquid is a cell growth media, which contains essential molecules for cell growth, and also sometimes growth factors and proper carbohydrate and oxygen and CO2. And this is a picture of a general typical tissue culture room. You can see this laminar hood, which ensures the no bacteria can get inside. So it has a laminar flow coming down, which is a filtered flow. So there's no bacteria, and you need to develop a procedure which will minimize any bacterial contamination. And this Petri dish is also prone to be, it's a wide opening, so it can be easily contaminated. So there are other types of dish. It's called a tea flask. So this tea flask has a much smaller opening, so it is minimizing the contamination. And this is a typical picture of a cell which is growing on top of the surface of this plastic. And these cells, you can see under the microscope, and they grow and grow. And we are using these grown cells for our specific experiment, typically in vitro experiment. So then we need to understand the terminologies more here. So in vivo and in vitro experiments, two different ways of conducting biomedical experiments. Let's think about an example of cancer biology. We want to study cancer to develop a drug which will kill specifically cancer cells. Then we need the material, cancer cells. The cancer cells are, some of the cancer cells can be very strong, that it's almost immortal, and we can grow cancer cells in a test tube over many, almost infinite passages. So this we call as a cancer cell line. And let's try to study, for example, breast cancer. Then you can find out breast cancer cell lines, which originated from each of the patient's aggressive cancer cells. So one cell line probably means one representing one breast cancer cell patient's cells. And this has been growing inside a Petri dish, and we grow them enough, and we have, let's say, candidate drug, which will supposed to kill this cancer compared to another control drug. So we can test them out. So this is one way of testing a drug, developing a drug, which is anti-cancer, let's say, against breast cancer. However, the problem is we can kill cancer cells very well in a laboratory environment, in a Petri dish. But why there are still many patients dying from cancer, while we can kill cancer cells very easily in this Petri dish. And there could be a lot of reasons for this. However, this environment is very controllable so that we can do a more detailed study. However, we have to always think, what's the reality? Why, when we give this cancer drug candidate to a patient, why many times it doesn't work? And then we have to think about, hey, does the drug administered to the patient get to the tumor site enough concentration? This is a drug delivery issue, and many times this becomes a big problem, which requires an engineering point of view of approach for this drug delivery issue. And on the other hand, this Petri dish environment might not be mimicking fully what's happening inside the body. So as an alternative, we develop a way of testing this drug in the living system, such as an experimental animal, for example, a mouse. So we can grow a human tumor growing in the mouse, and which is more likely to be closer to real human body. So then we give the drug to the mouse and see if this drug effect will slow down the tumor growth or even shrinking the tumor. And then we try to understand what's happening. And this type of study we call as an in vivo study. And of course, even if we can cure a tumor in the mouse well enough, but many times this may not be sufficient enough to guarantee that this drug will work in human, because humans is more complicated. But this is a way how in biomedical experiments we can conduct and develop a strategy how to treat or monitor the drug activity. So here's a table I prepared. So that's typical two approaches as in vitro experiment and in vivo experiment. So the meaning of in vitro means inside the glass, such as test tube or Petri dishes. While in vivo experiment means in vivo is the Latin and meaning that inside a living body or living organism or living animal. And they have both pros and cons, or advantages and disadvantages. So in vitro experiment is we can replicate this test tube many times, and so we can control it easily and we can replicate easily, and relatively costs are lower. And also faster, you can do experiments in weeks or maybe even days or weeks. And also these cells derived originally from even patient, but has been long time ago. And these cellular materials are causing less ethical issues. While in vivo experiments is you have to have an experimental animal. So the tumor, let's say tumor cells growing inside an animal body will probably make it more intact biological microenvironment. So that means the phenomenon, the results you obtain from the in vivo experimental animal body could be more relevant to the real human patients. So let's think about the limitation of these two approaches. In vitro approaches are, it's easy. You see the cells growing in the media, you apply a drug to see if how many cells are being dying. And however, this is not mimicking the real microenvironment. So all microenvironment cannot be taken into account. For example, like tumor growth requires a new vessel growth from the surrounding vessels, which we discussed as angiogenesis. However, in vitro system, there's already plenty of an oxygen and you don't even need to consider blood vessel. So, if you want to study this part, it's maybe not easy to study with in vitro experiment. And also, this controlled environment makes it easy to draw conclusion, but that study results may not fully represent what's really happening in the whole organism. So this gives a great insight. However, we have to always ask question whether the result from in vitro is truly realistic. And so we need to test it later on, being able to interpret as a more meaningful results. On the contrary, in vivo experiments is actually using a real organism or animal that is more difficult to control. And, you know, in science, repeatability is very important. So the result what we observe or we got can be reproducible from other laboratories, makes the result more reliable, and people consider it very important. And that is not as easy as in vitro experiment. And of course, instead of cell line, we are using a real animal, and that becomes more expensive and of course, time consuming. And also at the same time nowadays, ethical issues are important because animals, we have to protect them. We have to consider ethical issues of this experiment. So in these aspects, we have to consider which part to use. Usually people start to use in vitro experiments first, for example, to find out candidate anti-cancer compounds. And then from them, because in vivo is more experiment, those, maybe the best candidates, we can start to using in vivo experiment to see if they have more efficacy against these particular cancer models. And on the other hand, there are ex vivo term, which means out of the living body. So which, when we study of tissue, which is directly removed from an intact organism, and when we call this type of experiment, we call ex vivo experiment. For example, if people want to study brain electrical phenomena, and instead of using in vivo, which is maybe more difficult and complicated, you can just take out the brain from the animal and slice it and make a brain slice and do your experiment with this brain slice, which is kind of still alive. However, it's taken out of the original animal body. So that kind of experiment we call as an ex vivo experiment. And hopefully this makes you understand how to approach this type of biomedical science.